Chapter 2 Biological Classification NCERT full chapter. In this section, we deals about the general introduction about classification. Using simple morphological characters, Aristotle put forward the first attempt of classification. He classified plants as trees, shrubs, and herbs, while animals as those which have red blood and those that did not. Later, the Linnaeus put forward the two kingdom classification. In two kingdom classification, there are two kingdoms. One is Plantae and another one is Animalia. But he does not distinguish, Linnaeus does not distinguish whether they are unicellular, whether they are multicellular, whether they are eukaryote or prokaryote, unicellular or multicellular, photosynthetic or non-photosynthetic. Those characteristics are not considered by Linnaeus. So that is the two kingdom classification. Now coming to the next section that include a general classification. Here we can see the five kingdom classification. Five kingdom classification was put forwarded by R.H. Whittaker. R.H. Whittaker put forward the five kingdom classification. There are five kingdoms. They are Monera, Protista, Plantae, Fungi, Plantae and Animalia. So they are based on their cell type, their cell wall, their nuclear membrane presence or absence, their body organization, whether cellular, multicellular, tissue, organ or organ system, and they are based on their mode of nutrition. They can be autotrophic, they can synthesize their own food, or they can be heterotrophic, in which they depend on some other organisms for the energy. So based on these 1, 2, 3, 4, 5, based on these five characters or based on the five important characters, they are classified as five kingdom classification. It was put forward by R.H. Whittaker. Okay, all these characters are very important. This table is very important. So the table is very important. So here the Monera, they are prokaryote in which there will not be any membrane bound organelles. In the case of Protista to Animalia, all are eukaryote. That means they have a membrane bound organelles. Now the cell wall, in the case of Plantae, they have a cellulosic cell wall, but in the case of Animalia, there will not be any cell wall. So there will be a nuclear membrane in the case of all the kingdom from Protista to Animalia, except the case of Monera. And the body organization can be started from the cellular to the organ system in the case of Animalia. And the mode of nutrition include autotrophic to the heterotrophic in the case of Animalia. Now the kingdom Monera. In the kingdom Monera, the main important members are the bacteria. So based on their structure, they are divided into cocci, bacilli, spirilla, and vibrio. Vibrio are comma-shaped, but in the case of spirilla, they are spiral-shaped. But in the case of bacilli, they are rod-shaped, and in the case of cocci, they are simply a spherical-shaped structures. So Monera main members are bacteria. They are seen everywhere, from their handful of soil to the deep oceans, deserts. No, they can be seen everywhere. Okay. Here's the Archaebacteria and the Eubacteria. Archaebacteria mainly seen in the harsh habitats. So they include salty areas like halophiles, in hot springs called as thermostophiles, in marshy areas called methanogens. These methanogens are Archaebacteria that are seen in the guts of cows and buffalo, which can produce methane, a biogas. Now the Eubacteria, also called as true bacteria, and they are characterized by presence of rigid cell wall, and if motile, they will have a flagella. Now, one of the examples cyanobacteria, they are photosynthetic autotroph. They can synthesize their own food. And the examples are nostoc and anabaena. This nostoc and anabaena have a structure called as heterocyst, which help for the fixation of nitrogen. Now, there are chemosynthetic autotrophs in the eubacteria. They will use inorganic substances and they will oxidize these inorganic substances and produce ATP. Whereas heterotrophic bacteria, they will mainly include decomposers, and some can make the curd. We know curd, the curd from milk. The bacteria that coming under the heterotrophic bacteria can some of the members can synthesize curd from milk, and some can help for the production of antibiotics. Now the protista. In the kingdom protista, all single cell eukaryotics are placed in the protista. Their members include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. So the first one is chrysophytes. Chrysophytes include the members called as diatoms and golden algae. These diatoms have a shape of like a soap box shape. They have a soap box shape in which there will be two overlapping shells. So the diatoms have a cell wall that forms two overlapping shells which fit together like a soap box. They are mainly seen in the marine areas and they can they can synthesize diatomaceous earth. This diatomaceous earth is used for polishing, filtration and filtration of oils and syrups. Now this is the structure of the euglena which is coming under the euglenoids. So the second member is the dinoflagellates. Dinoflagellates can produce different colors like yellow, green, brown, red, etc. based on the pigment that is present in their cell walls, in their cells. So one of the example is goniolax. This goniolax will produce the red sea. They can produce the sea like red in color, also called red tides. So that is goniolax. And the third member is the euglenoids. They are very flexible because of the presence of a pellicle. They have a protein-rich layer called pellicle, which helps for their body to be flexible. One of the example is the euglenoids. You can see here the pictures is. Now the slime molds. Slime molds is here. You can see the structure of plasmodium. They will spread like an spread over a feet of areas. Okay, and they can also produce pores. These pores will have true valses. And they will survive in the highly extreme conditions in the by producing like a spore-like conditions. And when the conditions are very favorable, they will regain its structure. So that's about slime molds. These are the slime molds. Now the protozoans, all protozoans are heterotrophic and live as predators or parasites. Okay, they are predators or parasites. There are amoeboid protozoans, flagellated protozoans, ciliated protozoans, and sporozoans. Their features are defined here. The flagellated protozoans examples include trypanosoma. This is a trypanosoma. And the ciliated protozoans include paramecium. And the sporozoans include the plasmodium, which cause the disease malaria. It's a malarial parasite. So these are the examples of protozoans. So here we already completed the discussion of kingdom Protista. Now comes the kingdom fungi. The kingdom fungi, we all are familiar with fungi. From the we can see fungi in the bread, in the rotten egg, everywhere. Okay, so there are fungi that are very common. Fungi have the specialty like they can use the soluble organic matter from dead substrates. Hence, they are called as saprophytes. They are called as saprophytes because they absorb soluble organic matter from dead substances. They are called as parasites because they can live in the living plants and animals, and they will depend on the living plants and animals, hence they are called as parasites. And they can also form association with the algae, and thereby they can form lichens, or they can also form association with the roots of higher plants, and thereby they can form mycorrhiza. So they can be saprophytes, parasites, lichens, or mycorrhiza. So the plasmogamy, karyogamy, and meiosis are the three important steps coming in their sexual cycle. That is, plasmogamy is the simply the protoplasm, and karyogamy is the fusion of their nucleus, and simply the meiosis is the division of the cells into haploid spores. So that is the plasmogamy, karyogamy, and meiosis. These are the three important steps. Now, they are based on their morphology of the mycelia, mode of spore formation, and fruiting bodies, they have been divided into a different kingdom. So which includes different kingdom and into various classes. So this include phycomycetes, ascomycetes, deuteromycetes, and basidiomycetes. So the first one is phycomycetes. These phycomycetes are mainly seen in the aquatic habitats or in the decaying woods or in the moist damp places. And their mycelia means their main cell body is termed as mycelia. It is not partitioned by anything. They will be have simply they likewise this will be their mycelia. There will be numerous nuclei without any partition, that is called as choanocytic. So they are choanocytic in nature. There will not be any partition. They are choanocytic in nature, and their asexual reproduction is by zoospores or aplanospores. Zoospores are motile asexual spores, and aplanospores are non-motile asexual spores. So that is about its reproduction. And the zygospore is the sexual spores. A zygospore is formed by the fusion of two gametes. These gametes are similar in morphology, and that is called as isogamous, or dissimilar in morphology, that is called anisogamous or oogamous. And some common examples are mucor. Examples are include mucor, rhizopus, albugo, etc. The next member is the ascomycetes. Ascomycetes are commonly called as sac fungi. Okay, they are called sac fungi. One of the important examples is penicillium. One of the important examples is penicillium. Ascomycetes have conidiophores and conidia. Conidia are they are asexual spores. They are produced outside. You can see this conidia. When they have sexual spores called as ascospores. They are produced in a sac-like structure. So they have the asexual spores called conidia exogenously, whereas they have ascospores which are produced endogeneously in sac-like structure. So that's all about its structures. Now the basidiomycetes. Basidiomycetes is very important. All the fungi, which like the mushrooms, belong to the basidiomycetes. So are mushrooms, bracket fungi or puffballs. They grow in soil or logs, have tree trunks in living plant bodies as parasites. So they include rust and smuts are coming under the basidiomycetes. Mushrooms are coming under the basidiomycetes. Puffballs are coming under the basidiomycetes. So their sex organs are absent, but plasmogamy is brought out by fusion of two vegetative somatic cells at different strains or genotype. So the two strains will be come together and fuse, and they will form the diploid stages. And here we can see the diploid basidium and meiosis. They will produce as the basidiospores, and the basidiospores will in turn germinate likewise. This will be their life cycle. So their important members are agaricus, a mushroom, a euslagus mud, and Puccina is the rust fungi. Now the last member is the deuteromycete. They are called as imperfect fungi because they have only the sexual reproduction are known. Their asexual reproduction are only known. There is no sexual forms of these fungi are not yet discovered. If these fungi sexual forms are discovered, they will either move into the basidiomycetes or ascomycete likewise. So that is all. That's why they are called as imperfect fungi. And the deuteromycetes examples are Alternaria, Coleotrichum, and Trichoderma are the some of the important examples. Now that is the deuteromycetes. They have only sexual stages in their life cycles. Okay. Now we are in the case of some importance. Now the kingdom fungi, I already discussed. Now the kingdom plantae and kingdom animalia. Kingdom plantae, they have an alternation of generation in which they will have a diploid main plant body will be a diploid sporophyte, and they will produce the haploid gametophyte. This alternation together form the alternation of generation. They will be discussed in the later chapters. Now coming to the kingdom animalia. Kingdom animalia will also be discussed in the coming chapters. And at the end, that is the virus, virions, prions, and lichens. So what are called a virus and what are their important features? So virus, you are already know that coronavirus. Okay, so viruses are non-cellular organs. They are non-cellular organisms that are characterized by having inert crystalline structure. They have an inert crystalline structure. The meaning of virus is venom or poisonous fluid. It is a Dmitry Ivanovsky in 1892 first recognized the microbes as casual organism of the mosaic disease of tobacco. And later, the one of the M.W. Bejernik, M.W. Bejernik, he demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plants and named the new pathogen as virus and called the fluid as Contagium vivium fluidum, infectious living fluid. And the third scientist is Stanley in 1935 showed that viruses could be crystalline. So Stanley crystallized the virus, and the Bejernik demonstrated the transfer of the virus, and it was the tobacco mosaic virus was first causing organisms. So mosaic virus was first introduced by Ivanovsky. So there can be either RNA viruses are there and DNA viruses are there. So no virus contain both RNA or both DNA and RNA. Either it will be RNA virus or it will be DNA virus, not both together. They cannot be seen. So that's the virus. AIDS is caused by a virus, and in the case of the virus, they have a structure. They have an outer covering called the capsid. It is mainly made up of capsomeres. Subunit of capsid is capsomeres, and they will have either DNA or RNA. Now coming to the bacteriophages. Bacteriophages are the virus that attacking the bacteria. So bacteria attacking viruses are called bacteriophages. They have a head-like portion in which with inside the head we can see their nucleotide either RNA or DNA likewise, and there will be a collar-like structure and there is a sheath-like structure, and they have tail fibers. These are the structure of a bacteriophage. So viroids are first introduced by Dinier, and the prions are first the another one that has been causes some infectious neurological diseases, and it's been transferred by agent consisting of abnormally folded protein. For some of the disease caused here in animals include mad cow disease in cattle, and its analog variant CR Jacob disease in humans. A lichens is simply an association between a fungi and algae when the fungal component is known as a mycobiome and algal component is known as phycobion. So phycobion and mycobion together form the lichens. Do subscribe the channel.